We have three presenters, all of whom received funding from DFO for their research. We're going to begin with Noelle Mason. She is a third-year undergraduate at Colorado State University, and she's studying fish, wildlife, and conservation biology. She aspires to become a science communicator for conservation research endeavors, and her research involved gauging the life stress of some common songbird species. So Noelle, are you ready to go?
Yes, I am.
Excellent.
It is telling me that I can't screen share. There we go.
It'll be all good now.
All right, well, good evening, everyone. My name is Noelle Mason. I am a third-year undergraduate at Colorado State University, and I do undergraduate research, among other things, in Dr. Kristen Ruig's lab. This is a conservation genomics lab that does population genetics research on migratory songbirds, and I'm very lucky to be able to work with them. And I first want to thank the Denver Field Ornithologists for allowing me to use this money for my research. And if you can't hear me for whatever reason, I had Wi-Fi issues and ended up at Starbucks tonight. If you can't hear me or the music's too loud or something, let me know and we can hopefully try to get it resolved. So we'll hop right in, maybe if my screen will switch. I used the DFO grant for two primary things. The first was to establish a feeder watch program at Colorado State University's Environmental Learning Center, which is a 200-ish acre property to the east of us. And they have summer camps there and a bunch of other different educational opportunities for children in the community. So that's what I will talk about first. Around this time last year, myself and a couple of graduate students took some bird feeders out to the Environmental Learning Center and set them up in sort of a triangulatory fashion. So you can see, if you're standing in the middle of them, you can see all three of them at any point. And they are also, this is sort of a wetland property, the Environmental Learning Center. So we stayed away from the rivers and the swamps so the birds would have something, somewhere to land, and with bigger trees and all. So these pictures were taken in the spring and in the winter. Some of the favorite birds that come to stop at the feeders are, I personally love all the goldfinches. We get flocks and flocks of goldfinches every year. Well, I guess for the past 2 years now, and just huge numbers. And we also get our ground feeding birds. There's a couple of different goldfinches here in this center picture, a chickadee, and we also get quite a bit of juncos feeding from the ground, as well as some pretty cool woodpeckers that also join us throughout the year, and then amazing birds in the summer as well. We've had a couple of really cool birds join us throughout our field season. So we had a really gorgeous blue grosbeak stop by last summer. That's my hand, someone else took the picture. But he was a real treat to catch in our nets. And then there's also been, I think, some increased raptor activity. I'm not sure if that's a good thing, but it does seem to be a good spot to raptor watch. We got this textbook red-tailed hawk to look at also this year, as well as a juvenile indigo bunting that was really gorgeous, some really beautiful coloring on her. And then also just there's a ton of goldfinches and they just look obviously gorgeous when they change plumage. So we also use the Environmental Learning Center as a research and training facility. These are some graduate students in my department with some red-winged blackbirds that we caught. We set up our mist nets near the feeders because we know that that's where the birds are going to be. Most of the time we are looking for black-capped chickadees and red-breasted nuthatches. But I mean, there's a ton of birds that fly through there, so we get a variety of different things. This facility also is used as a training facility for bird banding. for myself and other undergraduates that are involved with the program, as well as some graduate students that are newer to the program. Oops, switch prematurely. We learn how to mist net there. So we'll learn how to extract birds from the net, set up the nets, take down the nets. And then also I got to do some banding last summer, which was really fun. Also nerve-wracking. You never know. You have no idea how fragile birds are until they're in your hand and you're trying to put something on their tiny little leg. It's really kind of scary. And then we also, I have not done it yet, but there are also people that are learning how to draw blood from birds for other DNA analysis. And on the far right here, you can see our PhD candidate, my personal mentor, Marina. She's teaching one of my friends and another graduate student how to extract a black-capped chickadee from the mist net. So the Environmental Learning Center is also a facility for learning, obviously, and education and outreach. Last fall, we were really fortunate to be able to host a couple of field trips for a couple of elementary school groups that are in the Fort Collins area, where they got to learn about mist nets. I don't know if you can see in this picture up on the top left here, but that's our postdoc, Sheila. She is behind a mist net, you can hardly see it, but she is teaching people how to take birds out of the mist net and that it doesn't hurt them. We have on the bottom right here a little bird watching station where Taylor, our master birder of a grad student, was teaching some of the fourth graders how to use binoculars. I have no idea what they were looking at, but it seemed like they were successful with it. And then on the top here, we have Marina teaching the fourth graders about migration and what a genoscape is. And I'll get into that a little bit later, but basically the main research goal of my lab is to create these genoscapes for several species of tropical migrants, where it links different genetically distinct populations to that we find on the breeding ground to their location on the wintering ground. And on the bottom here, we have a cute class picture, also with the gorgeous scenery of the ELC. It's really beautiful in the fall. So if you ever get a chance to come up to Fort Collins, it's a favorite place for local birders. We have the feeders there. There's all kinds of beautiful birds that fly through. So yeah, I definitely encourage you to check it out. You can hike. It's open to the public. I highly recommend checking out the Environmental Learning Center here at Colorado State University. So that was the first part of my research, the first part of my grant. The second part is going to get real sciencey real fast. So what I wrote my grant proposal on was this first title here, which is the rate of avian telomere shortening between blood and feathers. And that was what I was going to do during my research experience for undergraduates last summer. But I had a little difficulty and ended up switching projects, but I will give you the details on both of these here shortly. So we have to start out with a definition of a telomere. Telomeres are, I like to think of them as the little plastic bit on the end of a shoelace that protects your shoelace from unraveling. So telomeres are a non-coding stretch of DNA that is on the end of our chromosomes, which protects our DNA as our cells divide. So each time our cells divide, they shorten, and telomere length can shorten with, the length decreases with time and it can be accelerated by stress. Therefore, it can be linked to relative life history, lifespan, and provide a proxy for relative fitness. So what both of my projects aim to do is use telomeres as a measure of stress. So my first project was to understand whether telomeres shortened at the same rate in blood and feathers. So whenever we're doing genetic work on birds, we can get DNA from both blood and feathers. If we're doing anything that involves telomeres, we often use blood because we assume that it offers the most real-time snapshot of telomere length. I'm sorry, they're playing REM and now it's distracting me. but it offers the most real-time snapshot of the telomere length in the blood when we collect blood. So you can assume that it shortens in real time. I hypothesized that feathers would have longer telomeres than the telomeres that we find in blood, assuming they would be collected at the same time, because when a bird is molting and their feather pinches off that little bit of skin that's left in the end of the feather, that's sort of like a window in time about back to when the feather was molted, and we assume that the cells are no longer dividing within that feather. That's what I would have guessed had I been able to finish this project. But it would be advantageous to be able to use feathers as a primary way to capture DNA in the field because it's less invasive. It's less invasive, and you don't have to, it's less time consuming, it's less expensive. You don't have to be trained on how to extract blood from a bird because that's also very time consuming. So it would be very advantageous if feathers did offer a real-time snapshot, or we could at least pinpoint the telomere length back to a moment in time that's predictable across all birds. So that was what my project was aiming to do, was understand if telomeres shorten at the same length rate in blood samples versus feather samples. Something I had a lot of trouble with last summer was being able to get recaptured birds, the same individuals several times. We were able to recapture a couple of birds, but we had a hard time. I really wanted to have a sample size of about 30, so it was very difficult to get 30 chickadees were my main target, and house finches just because they were the most common there. But it was really difficult for whatever reason to get the same individuals to come back to the net multiple times. So we're hoping to continue this in the fall and winter. A different undergrad is probably going to take this over so they can continue it after I graduate. And we're expecting winter to be better for this because the birds are going to be more reliant as on the feeders as a food source. I should have probably started with that this was going to be conducted at the Environmental Learning Center because it was centered around these birds using the feeders as a reliable food source. So because I had some time constraints with this and had to come up with a different project for my research internship that I had last summer, I switched gears to something that was a little bit more short term, also using telomeres, which was understanding differential stress experiences in niche tracking and niche switching yellow warblers. So this bird here on the right, you guys all probably know as a yellow warbler. They're really super handsome little guys. And my mentor, Dr. Kristen Ruig, and a former postdoc of hers, Dr. Rachel Bay, published this paper last year about genetic connectivity in the yellow warbler. So across this range, this is a genoscape, by the way. This is the main goal of what my lab does, is to create these for 100 migratory species. And up here on the breeding ground where we have this color gradient, this depicts delineation between several distinct genetic populations where they've been selected for on the breeding ground. Down here in Mexico and Costa Rica and the very top part of South America, here we have points where samples were gathered on the wintering ground, and you can see the colors match up to the range on the breeding ground. So you can see that these birds that hang out here in the west with us in the summer spend a lot of their time down in Mexico in the winter, whereas some of the birds in the blue spend a lot of their time further down in Central America. So the birds that spend time with us here in the west in the dry zones, they also spend their winter in dry zones in South America. So these are known as niche trackers, meaning they track their habitat over the course of their migration. But some of these birds that live here in the dry west with us, they also are found down here in Costa Rica, where it's super wet and rainforesty. And those are niche trackers. And my goal is to understand which of these two groups experiences more stress over the course of their lifetime as they migrate. So something that I would guess is that the niche trackers are going, or excuse me, the niche switchers are going to have shorter telomeres because they experience more stress as they're going from two very different environments multiple times over the course of their lifetime. So I would expect their telomeres to be shorter because they've experienced more stress over the course of their lifetime. I'm going to answer this question using telomere length, and I'm using actually a direct subset of the Bay 2021 data, which I luckily actually already had in my lab, which was sort of the reason that I was able to switch so seamlessly to this project. And I'm picking 10 switching samples and 10 tracking samples and measuring their telomere length using quantitative polymerase chain reaction, or qPCR. You probably are familiar with PCR, at least from COVID-19. It's a method where we're able to chop up DNA and amplify various pieces of it so that it can be measured more easily. The Q part is just what allows us to compare it to each other. So once I have done PCR on all of the samples, we'll be able to compare them all to see which individuals has the shortest telomere lengths. I'm going to compare this using the linear regression, just a statistical model that accounts for actually several different variables that could mess this up. So I'm accounting for sex, age, and migratory distance, just to make sure that none of those things are disproportionately stressing a bird out, and we wouldn't be able to tell if the niche switching or niche tracking was what is making their telomeres short. So the implications of this, I was supposed to do the qPCR and figure out the lengths last Friday, but our primers didn't come on time, so I'm hoping to do that this week. And I am hoping to quantify the stress impacts associated with niche switching, possibly niche tracking, to reveal selective pressures associated with the wintering ground specifically. If the results support my hypothesis, we would assume that birds are experiencing some sort of selection on the wintering ground rather than the breeding ground. But if the results support the niche tracking hypothesis, we would assume that these birds are probably going to be not necessarily set up for success as the climate continues to change, and it would help us to illuminate the adaptive capacity between climate generalists and climate specialists, letting us use genomics for climate change, which is really the whole goal of Dr. Kristen Ruig's lab. So that is the long story of my research that I did last summer and this over the course of this school year. And I just want to open it up for any questions, and thank you guys all for being here, and thank you to DFO for the grant. I really appreciate all your time.
Thank you. Thank you, Noelle.
Thank you so much.
So, wow. That was a lot to cram into a very short presentation. And thank you so much for doing that, and for doing it under the adverse conditions of being in a coffee shop instead of comfortably in your dorm room. So kudos to you for making it work. So you answered one question I had right away, which was, can you visit the CSU Environmental Learning Center? And it sounds like you can. Is it open all the time, or I mean, on what kind of hours are there?
Yeah, it's open 365 days a year. I don't know I believe it's daylight hours only. I don't think you can go stargazing there or anything, just because it is also a learning facility. They don't want any hooligans there at night. So, but it is open 365 days a year during the day, and it's a really beautiful wetland area, riparian zone. So, and there's a ton of birds. Like during migration, right now is a fabulous time to go bird watching there. It
Great.
is gorgeous.
Great. Cool. A couple of questions have come in. One is, will the qPCR results be communicated to DFO, or is that outside of your funded research? Would you be interested in sharing that with our audience?
Yeah, I
Regardless?
definitely would be interested in sharing that with you guys. I'm hoping to have it actually finished by the end of the school year. But the mail, the primers, hopefully it kind of is dictated when the primers come. But yeah, once I finish that, I'm definitely open to sharing that with the DFO audience.
Great. I will make sure I connect you up with one of our writers for our
Sure.
newsletter.
Great.
And then when you're testing the feathers, do you physically remove a feather from the bird?
Yes.
And how do you decide what feather to remove?
Yeah, so there's actually sort of a standard protocol for removing feathers. We always take a feather from the tail, and we take it directly from the center of the tail, because it's probably going to have the least impact on the bird there. If it appears that when you have the bird in hand, it seems like they've lost multiple feathers, we just won't take any of them, because we want to be as least invasive as possible. So when the bird has enough feathers, we'll take it directly from the center of the tail.
Excellent. And one last question. Does the grant money what was the main thing that you used the grant money for? Was it for supplies or for paying assistance or?
Yeah, so it was for all the genetic analysis tools. So for the primers, for the qPCR, as well as initially we had some set aside for extraction materials to extract DNA from blood and feathers from my first project, which I wasn't able to do, but those materials will go to someone else that's going to do the same project.
Great. Super. Well, thank you so much for your time. And I know you have to get off to the next thing in your evening, and we really appreciate you being here.
Awesome. Well, thank you all for having me. I really appreciate it. I'm sorry I can't stay to see the rest of the presenters, but I know this is being recorded, so I look forward to watching it another time and connecting with everyone else that way. So thank you so much for having me. I really appreciate it.
All right, so our second speaker is William Anderson. He is a master's student in the Taylor Lab at CU Boulder. His work focuses on hybridization and the speciation process, using avian hybrid zones to begin to understand those processes. He's currently studying the hybrid zone between the divergent lineages of house wren along the Front Range of Colorado. William, you're up.
Awesome. So thank you so much for that introduction, Sharon. So I'm going to be talking about house wrens in Colorado. So you guys are all very familiar with house wrens. I'm assuming pretty much everyone has them in their backyard. But what you may not know is that these house wrens are actually genetically ambiguous. They're actually hybrids all throughout the Front Range. They're very fascinating birds. So yeah, I'm going to be talking about characterizing their species boundaries and characterizing the hybridization in these birds. So to begin, I want to first give a background on just the speciation process itself. So here we have two very basic examples of speciation. On the left, we have the Hawaiian honeycreepers, which many of you are probably familiar with. They all came from one ancestor that colonized the Hawaiian Islands, and they all radiated out into different niches because they were different open niches. In the same way, the cichlids in Africa were in different lakes, and then they eventually radiated out due to just niches being open and niche availability. So generally, speciation occurs through 4 different mechanisms. The first is physical isolation. which is exactly what it sounds like. It's called allopatry if you're talking scientifically, but it's when a population is separated out from the parental population. And then two things generally act on it, which are genetic drift and natural selection. And both of those things move to change allele frequencies in the genome. So they change what specific traits are occurring. So you can kind of see this in like the Hawaiian honeycreepers here. You can see that a lot of them have that thin bill, but a lot of them have the thin bill to a different length, and they have different colors. So those are all results of drift and selection. And then the last thing is sometimes when populations come back into contact, there can be gene flow between populations. So generally, this is a very good graph of kind of how allopatry works. You have that original population on the left, the geographic barrier, and then while in physical isolation, reproductive barriers occur, and those are the ones that are caused by genetic drift and selection. And then you have these two populations that may or may not be capable of interbreeding with each other. So one of the best ways to study speciation is through hybrid zones. They've been called natural laboratories for studying speciation. This is generally because hybrid zones, at least in North America, are the result of what we call secondary contact. So about 10 to 15,000 years ago, there were glaciers all over North America, coming down to about the contemporary border between Canada and the US, and that forced all the habitat south. And so all the birds that were there were also forced south into what we call glacial refugia. So here on the screen, you can see the bullocks and Baltimore orioles, which is an example of this. But we also have things like the flickers, indigo and lazuli buntings, and the Futicus grosbeaks, so black-headed and rose-breasted grosbeak, all occurred during this time. So all of them have different levels of reproductive isolation. As you guys probably know, all of our flickers are pink along the Front Range. We have these intergrades between yellow-shafted and red-shafted. They're pretty much ubiquitous across the Front Range because there's a very low level of reproductive isolation there. That's why they're considered one species. Then if you move further up in terms of divergence, you have the indigo and lazuli buntings, which are much more divergent. They're considered species, and they don't really hybridize unless they're at range edges or if there's individuals. And then lastly, you have the Futicus grosbeaks, which is that rose-breasted and black-headed, and they're very different. They very rarely hybridize. And all of those are thought to have occurred in the same event, in that glaciation. So another species that is thought to have diverged during that glacial event was the house wren. So there are actually 32 recognized subspecies of house wren across the Americas. They go from Washington all the way to Maine, and then they go all the way south to Tierra del Fuego. So it's the largest, most widespread songbird in the Americas. And like I said, it has 32 recognized subspecies. About 5 of those are currently they're proposed to be split off because they look very different, but those are all island forms. But that still leaves 27 recognized subspecies, which is a crazy amount of subspecies. I focused on the 2 in North America because they were the closest to me and the most easily accessible. And they also have a lot of interesting questions to be answered. So we don't actually know their subspecific ranges. We know that there's an eastern and a western because the ones in the east look a little bit different than the ones in the west, but they pretty much sound the exact same. The only thing is there could be a call difference, but we don't know. It's never been documented. So they're very, very, very visually and vocally similar, but in their mitochondrial genome, they're about 5% divergent, which is the same as black-capped and Carolina chickadees. So those are 2 different birds that we consider species, and house wrens are that divergent in their mitochondrial genome. And the mitochondrial genome also says that they're non-sister. So that means that there was more divergent branching between these species. So they're relatively different, but we still consider them to be one species because they look and sound the same. But there's been no nuclear work done, so I really wanted to change that. So yeah, my goals were to define the geographic boundaries of these 2 subspecies. We wanted to find the western edge of the eastern bird and the eastern edge of the western bird, and then also detect and determine the extent of hybridization between the subspecies. So in order to do that, we first had to sample birds. So the way that we did that was through 3 approaches. The first was sampling individuals from the Boulder chickadee study. So the Boulder chickadee study is a nest box transect of about 400 boxes, and about 75 of those boxes are generally taken over by house wrens. So we had access to a really dense sample along an elevational gradient of house wrens. So we sampled 38 birds from that. We took blood samples from the wings. we then augmented that with 72 individuals that came from museums. So we had individuals from the Burke Museum at University of Washington. We had individuals from the Denver Museum of Nature and Science, and we had individuals from LSU's Museum of Nature and Science. We finally rounded our sampling distribution out with 18 individuals from museums, 9 of which came from California to represent a pure parental population from the West, and the Eastern population came from the East Coast to act as a parental population to anchor our samples. So these are our sampling locations. You can see it's mainly around the metro area, but it also extends out to central Nebraska, southern Colorado, and Wyoming, but mostly focused in the metro area. And then these were our nest box transects. So as you can see, there are 6 sites, of which we sampled 38 birds across about 15 nest boxes at each of these, across these 6 sites. So after we'd sampled all these birds, we had all of the blood, we then extracted them and sequenced them. So the extraction was done through a salt-based extraction, which basically just pulls the genome out, and then we sent it to New Jersey to be sequenced. After we had the sequences, we then ran something called a PCA. So a PCA is a principal component analysis, and it examines the genetic structure of populations. So it will show you if there are distinct genetic groups within the samples. And that's kind of how we would tell if both subspecies are present here. The next analysis we ran was called GG hybrid, which calculates hybrid index. So this is a little bit more complicated, but it basically tells you how much of the genotype of the target individual assigns to each parental population. So if you have an individual with 0.5 of hybrid index, that means it has 50% of its genotype comes from an Eastern bird and 50 comes from a Western bird. And it breaks down into any sort of fraction. So if you have a 0.19, that means it's 19% Eastern and 81% Western. So the results of our PCA show that not only are there Western birds here, but there are also Eastern birds here. You can see the Eastern is in yellow on the left, and the Western bird is in blue on the right. And you can see that some of the gray dots, which come from our sampling zone, they get close to those Western and Eastern parental populations. And this is backed up by our GG hybrid plot, which shows on the left with a value of 0 and in blue, the parental Western, and on the right in red with a value of 1, the parental Eastern. And so all those gray dots, those solid gray dots in the middle are individuals that we sampled. So they came from either the museum or the chickadee study. And something to note here that's actually very important is that the open circles are not individuals, they're just associated with individuals. So these 2 here on the left are associated with the leftmost 2 pure contact zone samples that we analyzed. And these 5 here are associated with these 5 down here. That means they came from the Boulder chickadee study. They came from those nest boxes that we sampled. And so what this means is that we know the birds sampled in those nest boxes were breeding. So we know that we have pure Western birds, all represented by these about 10 individuals. And we also have pure Eastern birds in the transect. This transect is about 14 kilometers long and covers about 3,000 feet of elevation change. But the fact that we have both parental types, as well as you can see, pretty much the whole range of the hybrid zone across our 14-kilometer transect means that there's some level of reproductive isolation occurring, that the hybrids have some kind of negative fitness effect because they're not persisting out into the population. Not everything is a hybrid. If there was nothing selecting against hybrids, you have that flicker situation where every bird is a hybrid. We can clearly see that not every bird is a hybrid. There's some Western and there's some pure Eastern birds. So within the bounds of the grant, we found that both subspecies are present along the Front Range. That was the first thing we wanted to do. We established that western edge of the Eastern birds and the eastern edge of the Western birds. It's right here along the Front Range, which is very cool. We also found that there's extensive hybridization between the populations. It's what we call a multi-generational hybrid zone. But moving forward, we were really interested to see what's facilitating the hybridization between these birds. So we know about 2 things, or we have an idea that potentially these 2 things could be interacting to affect hybrid index. So elevation and habitat partitioning. So elevation, what we expected was that house wrens would match other bird systems in this area. So as you guys kind of know, we'll find more pure red-shafted flickers in the mountains, but we won't really find pure birds in the plains. That's because, and it's the same for warbling vireos, you'll find mostly eastern warbling vireos in the plains, but you'll also find some Western, but you'll only find Western in the mountains. And it goes that way for a lot of different things, hairy woodpeckers, downy woodpeckers, all those things. The Western birds are capable of coming out of the mountains, but the Eastern birds are not capable of going into the mountains. And so our expectation was that would be the same in the house wrens, but it was not. We found an individual with a hybrid index of 0.58 at 8,300 feet. So that means that it was 58% Eastern at our highest sampling site, which is not what we would expect at all. And so we wanted to try and explain that. And so what we came up with so far is that it has to do with habitat partitioning. So on the plains, these house wrens are mostly using riparian systems. So they're using thickets. And so in the mountains, the only place where that habitat exists are along creeks. And that's where all of our boxes are placed, because generally the roads follow the creeks. And the Boulder chickadee study is actually targeting hybrids between chickadees. So the best place to find hybrid chickadees is along these riparian systems where black-capped and mountain chickadees can come into contact. But if house wrens are following the same system, then we would get an increased amount of hybrid or Eastern house wrens, which would it would mess with our results. Because what we did is we ran a regression to see if there was an association between elevation and hybrid zone and hybrid index, which is what we would expect. We'd expect more Western birds as we got higher up and more Eastern birds as we moved further out into the plain. And we found a non-significant result, even though the eyeball test shows that there is a significant correlation. So moving forward, what we've done so far is establish subspecific boundaries of these two birds, and we've also characterized the hybridization between these two subspecies. But what we want to do now is move forward and find the mechanism underlying this reduced hybrid fitness and see what exactly is controlling for these house wrens to be separated out. I would just like to thank everybody from the Taylor Lab. I'd like to especially thank Garth Spellman at the Denver Museum for all of his sample contributions, University of Washington, Louisiana State University, and all of you for your generous donations that make this grant possible. Thank you very much.
Thank you, William. That was amazing. I think it's one thing that I picked out just because I spent my last 7 years of my career in a collections museum is that the collections ended up being useful for your research. And that's something that we don't always know when the collections are collected, when the specimens are collected. Could you talk a little bit about that?
Absolutely, yeah. Specimens are an incredibly valuable resource because specimens, what we used the specimens for was just increasing our sampling size because we didn't have the funding or the time to extract samples from all of these birds. We only really had time for the 40 or 38 birds that we extracted. But since we had all these tissue samples from the museums, we were able to get rapid genetic sequences from, it was about twice as many birds as we took from our field sampling. So we pretty much tripled the size of our study through these birds. So yeah, it's just an incredibly valuable resource.
Great. So here's a question. How long have Eastern house wrens been here in the Front Range? I'm thinking of how settlers have changed the semi-arid habitat into a more eastern habitat, sort of like the incursion of blue jays into the Front Range. What do you think about that?
Absolutely. Yeah, that's a great question. So personally, I think that they probably have been here for a while just because they were able to follow those riparian systems. So they're probably following the Platte and the Arkansas. I think that we probably increased their prevalence. We've increased blue jay prevalence. We've increased black-capped chickadee prevalence. I think that these Boulder itself is just an incredibly riparian area. So yeah, we've probably increased their presence, but I think they've always been here, especially because they're in the mountains. We see them at 8,300 feet. So I would be surprised if they moved here and then pretty quickly moved into the mountains. I think that's probably been a rather established thing.
Mm-hmm. Mm-hmm. Great. Let's see, seeing no other questions, I am going to thank you for your time for this great presentation. I had no idea that house wrens were such a big deal, that there were 32 subspecies and they all pretty much look and sound the same. So that's pretty amazing. So thank you so much for your presentation.
Of course, thank you.
All right, so our last speaker this evening is Patrick McGee. He is an associate professor of wildlife and conservation biology at Western Colorado University in Gunnison. He teaches ecology, biodiversity, wildlife ecology and management, wildlife techniques, mammalogy, ornithology, and Colorado ecosystems. And he has been doing that for 25 years. He creates experiential field-based classes. In addition to teaching, he's dedicated to the conservation of local species and their habitats, especially the Gunnison sage-grouse and the sage-grouse ecosystem. Thanks, Patrick. You can take it away. We are seeing your email inbox. I don't know if that's
Okay. You want to see that?
we can answer your email for you if you want, but
How does that look?
that's the one.
Are you seeing the notes?
We're seeing the notes, yes.
Okay, let me try this over again. Sorry, it worked last time. Let's see. Now what are you seeing?
Looks good.
Okay. Back to the notes.
That's the notes again.
Geez, I don't know what I'm doing wrong here. Okay, try one more time. I think I know what to do. Now what do we got?
Yeah, it looks like your slide right now. That's it. You did
There
it.
we are.
And no notes?
No notes.
Okay. All right. Well, thank you so much for having me here tonight. And I really appreciate that nice introduction. My name is Pat McGee. I'm from Western Colorado University, as was mentioned. And I just wanted to say that I'm sort of filling in for my students who applied for the Denver Field Ornithologist Research Grant. And we're just so grateful for your support. And my student Ellie Orr, who's a graduate student here at Western, she completed her master's last year. And I had a second grad student that was also on that grant named Jake Bartholomew. And so he was also part of that. This study is going to be, this presentation is going to be a little bit different from the previous presentations. This is a very management and conservation oriented project. And it's kind of a cool example of just adaptive management in real life, in real time, where we're able to work in a collaborative conservation arena and collecting data, doing science to inform real-time conservation policy decisions. There's a group called the Slate River Working Group, which is up in Crested Butte, Colorado, along the Slate River, where the great blue heron colony exists that we're studying. And this group is created a management plan, and they're really looking toward the scientists and the observations coming from the field to drive that management plan and adapt that management plan as it goes along. So it's a pretty exciting project to be a part of, and looking at how science can inform management and how the work of the students is really important to that overall process. So we're looking at a number of different things, but I'm going to focus this talk on the phenology and the demography of the great blue heron colony up on Slate River, as well as the core question of our project is how human activity is influencing the heronry. I did want to, in addition to acknowledging and thanking you all for your generous grant for this research, also acknowledge Dan Paquette, who is a former student of mine and now lives in Oregon, and he is known as the Mindful Birder, and he has some absolutely beautiful photography, bird photography, but a lot of these pictures of herons in this presentation come from Dan, and I will do what am I trying to say? Anyway, mention that these heron photos are not Colorado herons, but these are Oregon herons. So anyway, they're beautiful heron pictures, but they're not my herons or our herons. So yeah, thank you so much. And we'll jump into this. One of the really neat things about great blue herons is it's not a real high priority conservation species, but it has some unique features that make it quite susceptible to human disturbance. And one of the things that we're just starting to learn more about every year as the population in Colorado increases and as the number of people are going deeper into the wild world of Colorado, that human recreation and wildlife, which historically have sort of been thought of to be able to coexist quite happily, and that human recreation has a neutral effect on wildlife, we're starting to realize that that is just not true. And there's a lot of impacts, and that we're going to have to make some challenging choices, I think, as we move forward in terms of thinking about how to manage human recreation in the context of birds and of other wildlife. But these herons are so interesting because they have this sort of dual personality where they spend the breeding season in this colonial nature, all together up on the treetops in their stick nests. And then they spend much of the rest of the year as solitary individuals, actually defending territories and spending a long time sitting in the water, fishing, waiting for that meal to come to them. I really have a fascination about these herons and their relationship to sticks. This photo is kind of nice because you can see these long legs of the heron and the 3 forward pointing toes, and then that one hind pointing toe that is a long toe. Of course, the sandhill cranes have a lot of superficial resemblance to herons, but when you look at the feet of the cranes, they don't have that long toe. It's a very short toe that's actually elevated off the same plane as the forward pointing toes. But what this foot arrangement allows is for these herons to grip and to live up in the treetops and to be basically perching birds, at least for part of the year. And they also just have this deep relationship to sticks, where they're picking up sticks for their nests. They're picking up sticks as gifts to their partners. Sticks are a very important commodity in the life of herons. But this foot has a big role to play in that lifestyle of caring about sticks so much. So anyway, these herons are up in the colonies in the tops of trees where they spend about 5 months occupying these nest locations, raising their young. They have a really strong fidelity to these sites, and come back to the same site year after year. It's important that they have a site that has the adjacency to food and foraging areas, as well as the right kinds of trees. There are herons that don't nest in trees, but most are treetop nesters. And the other key element to the appropriate nesting location is having some sort of isolation from disturbance, whether it's predators or human activity. So herons are pretty vulnerable to human disturbance, and most of the research out there from many decades of studying these show that herons do not do well in the face of humans, and they need this spatial buffer to keep them safe and to reduce the kind of abandonment of nest sites or reduction in reproductive success. So anyway, herons are not a real high priority conservation species, but when you think about this whole breeding biology, that you have all the eggs in one basket, so to speak, and compared to something like a mallard population where the nests are all spread out, if one mallard is disturbed, it's not the end of the world for that population. But if one colony is disturbed, it could have very important impacts on a whole regional population of this bird. We're really interested in the phenology of the great blue herons up at the Slate River and how they move through their annual cycle. Of course, during the breeding season, it starts with spring migration and arrival, and then the beginning of nesting and nest building. Corresponding with that is courtship and mating. This is the time when the herons are just so beautiful and their plumes are blowing in the wind up in 80 feet above the ground, doing all kinds of really elaborate displays. And then they start laying their eggs. They incubate for about a month, but they don't have a synchronous hatch. So the birds are coming out at different times and are different ages in the nest. And then they go through a relatively long 7 to 8 week brood rearing period to raise those chicks. And during that time, the adults are spending a lot of time out fishing and foraging and trying to provision these chicks. It's a very strenuous task for sure. And then the birds leave. And it's really fascinating. What we've seen is that as soon as the last chick leaves the nest, and within each nest, but also the colony overall, the birds basically do not stay in the area of the nests of the colony. And we really have no idea where these birds go after the post-breeding dispersal and then later in fall migration. So big mystery there for us. So our study really focuses on the effects of human disturbance on the herons. And there's a number of different ways we can measure that heron disturbance response. And the things that we're doing is looking at phenology, or how the herons move through their breeding season, all that annual cycle, part of the breeding season, part of that annual cycle. We're also looking at and have looked at the behavior of herons and how human activities is altering both their nesting behavior and their foraging behavior. And then we're looking at the demographics of the population and looking at how population size, the number of active nests, and the reproductive success are potentially being impacted by human activity. There's other ways to assess human activity, or to assess heron disturbance in addition to these, including nest abandonment, which we haven't seen, and also physiological indicators like stress and increased disease, which we are not measuring at all. The photo, or the little image here, shows these people on sup boards. What's interesting about this site is that about 7 or 8 years ago, there was very little human activity on the actual river in our study area, but that has picked up dramatically to the point in about 7 years ago, we had hundreds of people on the river, and the river goes directly under the nests. So there's no spatial separation between the human activity and the nesting herons. So that's what brought the what's called the Slate River Working Group together, led by an organization called the Crested Butte Land Trust. They came to us and asked us if we could start addressing this question and provide the science to guide the management planning and the Slate River Working Group policies. So here's our study area. This is the Slate River right outside of Crested Butte, Colorado. It's about 30 miles north of Gunnison, where I am and where Western Colorado University is. And this is looking out, the blue line there represents the actual Slate River. And then there's 2 colonies, 2 sub-colonies. What we call this the lower colony, and then this one the upper colony. You can see this lower colony has a bit more live trees. The upper colony is looking a little shabby. And what's interesting about this, somewhere I found it, I've never been able to repeat finding this information, but this is potentially one of the highest heronries in Colorado, if not the highest. And it's also unique because these trees are actually lodgepole pines. And it's strange to see lodgepole pines down here in the floodplain of the Slate River, but it's that's what it is. And you can see that those trees are, at least in the upper colony, are beginning to age, and these trees are actually younger in the upper colony than the lower colony, but they have more wear and tear, and I assume the herons have been in that part of the colony longer than in the lower colony. So this is our study area, and we are observing these birds from back here behind the colony, and from here, this is the Slate River Road, and then from this location where the photo's taken up on Smith Hill. I just throw this slide in super quick to show that we got this data from Colony Watch through the Bird Conservancy of the Rockies, and we really don't have much historical data on this colony, how long it's been there, how long it's persisted, what the numbers are, except a few points that came out of Colony Watch. And the data aren't that robust. These are probably one count a year, at a certain time. You can see the dates there. So not real robust data, but it's interesting to see, especially in 2010, that there were 19 active nests that were identified in the colony. Here's what we found over the last, we started this study in 2018, so this is our 5th year of the study and supposedly our last year of the study. But this is the number of nests, and the blue is the total active nests, and the orange is the total successful nests, meaning that at least one egg hatched out of those nests. And what we see is 25 nests in 2018. We went down to 20 total nests in 2019, down to 18 in 2020. And last year we had 16 active nests. So far this year we have 12 active nests, and there's still a chance for a few more birds to show up. But we've seen a 36% decline in the number of active nests over that 4-year period. So not a good trend. And we don't have real good, it's just correlational data, right, with the human activities versus any kind of experimental data. But these photos below are just showing the location of the nests within the lower colony on the left and the upper colony on the right. And here's just showing some of that demographic data across the 4 years of the study. It's interesting to see when these birds arrive in the middle of March, and on 3 of the 5 years, they arrived on the same day, March 13th. So they're almost as predictable as the swallows out there in Capistrano. But these birds sometimes, depending on the year, for example, in 2019, there was 2 feet of snow on the nest when these birds arrive. And so it's very interesting that they still come. The river could be totally frozen. There's not much foraging opportunity, but they come, and they've got a long breeding season. So the earlier they get there, the better in terms of getting things going. Some other things just to point out here, we had a total adult population during these years is 50 birds, 38, 34, and 32. The total number of chicks being produced, and then the number of chicks fledged out of those chicks. So you can see pretty high reproductive success, even though the total number of nests seems to be declining. And then here are the brood sizes across those 5 years. Pretty consistent, even though we're seeing fewer nests. The mean brood size remains pretty consistent. And then the range of brood sizes from 1 chick per nest up to 5. One of the main things we did, and this is just showing data for last year, but is do hour-long observation bouts to assess how much activity is in the area of the colony. Almost all of these activities are more than 300 meters away from the colony, but you can see the main thing are passenger vehicles along the road, making up 65% of these human activities. Bikers make up 16%, runners and hikers 8%. The number of, this isn't even on this table, but the number of river recreationists is much, much less than 1% of the total human activities. That's the only activity really, besides skiers, which we didn't have any last year, that are within the 300-meter buffer of the nest. And while it makes up less than 1% of the human activities in the area, somewhere between 70 and 100% of the heron disturbances are associated with river recreation. The way we started assessing the effect of river recreationists on the herons was doing time budget analysis. And we did pre-disturbance time budget, scan sampling, and then during disturbance, and then post-disturbance. So the researcher would watch the colony as some kind of boater was coming toward the colony, do a quick scan of every heron in the colony and look at its behavior, instantaneous behavior. And these graphs are just combining all of the non-disturbance behaviors and then all the disturbance behaviors. Disturbance behaviors include things like flushing, hunkering down in the nest, or being alert. So you can see pre-disturbance as before the boaters get to the site, 100% undisturbed behaviors. As we get into during the time when the boaters are in the colony stretch, we get about 52% undisturbed and 47%. These numbers were quite higher for disturbed behaviors in 2020 and 2019, more like 80% disturbed behavior. And then post disturbance, we have they go back to 100% undisturbed. So really strong evidence for a strong effect on heron behavior, with many flushing events occurring during these disturbance periods. This is kind of showing some of we call these floater events, and some of the characteristics of these floater events. You can see we just had in 2021, we only were able to observe 8 floater events. And partly that's because of the policy to have what we call a no float period during from March 15th to July 15th. So it's a voluntary no float period. So some people actually do float down the river during that time. And it's hard to always be out there. So we had in 2019 and 2020, we had researchers out on the site pretty much 7 days a week for several hours a day, and still weren't able to really capture that many disturbance events. So our sample sizes are pretty low. But when you look at 2019 and 2020 and 2018, our sample sizes are increasing, and we're basically seeing the same thing that we see in 2021, which is whenever humans are right below the nest, we see a fairly significant disturbance response by the herons, including a number of flushing events. But these are just ways, and I won't go into it just because we don't have that much time, but we're also trying to characterize these floater events in terms of how floating happens, and perhaps there's some human activities and protocols that would reduce the disturbance to humans. I don't think it has a big reduction because it's just that close proximity of humans to the herons. It doesn't really matter that much what the humans are doing, but just being that close creates quite a disturbance. And we have quite a few people that are not following the protocols that are floating through the heron stretch. So there could be improvement on education, but again, I don't know how much difference that'll make in terms of the heron response. So here is just kind of tying some of this adaptive management together. And when we look at the 4 years of the study so far, we look at river flows, and they were low in 3 out of 4 of the years. 2019, we had really high water, and in fact, during that period of June 6th to July 14th, the Gunnison County actually closed the river because of danger. So it was a regulatory closure, which was nice for the herons because it kept all the people off of the river during that time. And then the day before, so our voluntary no float again goes from March 14th to July 15th, or March 15th to July 15th. And that wasn't established, the voluntary no float wasn't established in 2018, but in 2019, we didn't really get to test that because of the regulatory closure. And then in the last 2 years, we've had relatively low water, and really the last date that someone could float the river was early July, before the non-floating period ended, which didn't make the river recreation community happy because they basically didn't have any opportunity to float. So some people did go ahead and float during the voluntary no float period. It was voluntary, so it was legal to float. But anyway, these are some of the numbers that we had that we were able to observe. And here's percent of events that led to heron flushing, 100%. And during disturbance, percent of herons in disturbance behaviors, 50 to 80% of herons were in disturbed behaviors during those times. So just to look at, I think I better check this chat. Okay, just want to make sure I'm not going too long. One of the really interesting things about these herons, once a colony gets below 20 nests, there seems to be quite a bit of evidence that they don't persist nearly as long. But what makes a site very suitable, and again, you have to have the right kind of habitat, the nest tree habitat in good condition. You also have to have the adjacency of foraging habitats. And then you need that third component of solitude. And when we look at the Slate River site, we've got, this clicks, we do have good tree habitat, even though some of those trees look like they are declining. When you look at the rate of decline, my student Jake did a lot of, did a whole study on this and found that these trees will still probably be persistent for many decades, even though they're starting to lose their branches and so on. There's also pretty good foraging habitat. This is a high elevation, sort of a oligotrophic system, so probably not the highest density of fish and so on in these wetlands and in the stream, but still pretty pristine area. The thing that's missing is the solitude. And so what we really need moving forward is if we want this heronry to persist, if we even have control of that, then we really need this spatial buffer of at least 300 meters, keeping the river recreationists away from those nests, especially throughout this whole breeding season, which goes from March 14th all the way till about August 14th. So that's the missing part. And right now we have a compromise going on, allowing for people to be on the river after July 15th, when you still have about 50% of the nests are active. And we also have only a voluntary no float period, and many people aren't really willing to sacrifice their opportunity to float. So I'll end it there. And I just again wanted to thank you guys all for your really generous support to our students, as well as these other entities. The last line here shows the names of the students, Jake, Bartholomew, and Ellie, or we're both grad students, then Megan Zariba, Cam Utes, and Hadley Nelson, and oh, I forgot to put Grant Campbell on there last as well, were my undergrad students working on this project. So thank you very much.
Great. Thank you, Patrick. We have a few questions here. I have always wondered what the survival benefit of colony nesting for such a large bird with few predators.
Yeah, I mean, it's they are by size able to be more resistant to predation, but they still have quite a few predators, especially bald eagles, and not just the birds themselves, but especially the nests. Birds like ospreys and other birds of prey can be nest raiders. Ravens as well can be nest raiders. So just being together, the basic idea is that you've got a lot of eyes on predators, and you can have more surveillance and just more ability to see those predators come in. And herons are pretty good at defending their nest if they're there. So, so anyway, that's probably the main benefit of having a colony versus a single nest.
Yeah. Next question, how is our severe drought affecting the herons?
That's a great question. I probably don't have a great answer for you. Certainly, as I was talking about the suitability of a site, that it's not only the colony trees, but it's the adjacency of good foraging habitat. And these wetlands are definitely impacted by lower snowfall. And, you know, the Slate River wetlands are a really pristine area that a lot of people have spent, you know, decades trying to protect. The Crested Butte Land Trust has this all in conservation easements and some owned property. But they, you know, there's beaver ponds, there's old former stream channels, there's all these different kinds of complex wetland habitat conditions. And so without good water flow, you just have less habitat within those systems. The herons are amazing though. I mean, even with this really cool wetland complex just at their feet, they don't spend a lot of time right there foraging. They move long distances. And we have another heronry that is down closer to Gunnison. And some of our fish biologists from Colorado Parks and Wildlife and the BLM did a study where they put little pit tags in brown trout. And then they were able to follow these pit tags to their destination, which they found many of them at the base of the trees in the heron colony. And these birds were flying, probably 15 to 20 miles to access those fish and bring them back to the colony. So herons, it takes a lot of energy. It takes a lot of time to provision the young and to catch all that fish. I know, just being a fisherman myself, but not a good one, how hard it is to catch a fish. And I can imagine just what a challenging job it is for them. And they have to move long distances to get that food.
And I'm not sure what the drought status is in the Gunnison River Valley there, so it may not be quite as bad as it is here in Denver.
It's not good. This year we have about 106% snow pack for at this time of year, so that's good news. But it's definitely been, as you saw those tables, the last 2 years have been pretty dry. And we're mirroring a lot of what's happening across Colorado and the West right now.
Next question, is there any opportunity to buoy off the rookery section or prohibit paddling in the area for the nesting season? And I know from being up there that there are wonderful signs that communicate to people the no float period, so that I think what this person's asking is there a way to up the game on really prohibiting people?
It's a great question. And, I think what we're looking at is this opportunity for collaborative conservation, which is a hard process, where you try to bring all the stakeholders together. That's what the Crested Butte Working Group is a really diverse stakeholder group. And so the river recreationists are at the table, along with the wildlife enthusiasts and the heron protection organization and the public land agencies and so on. But trying to come to a shared vision of how the river should be managed. And, you could just, it's challenging to just cut off the river. And for example, the Bureau of Land Management is not interested in doing that. There's also this long legal precedent about floater rights and water rights and all that kind of stuff. So it gets into some murky, very challenging complex legal questions. So nobody really wants to go there and just get caught up in some court battle for a long period of time. But on the other hand, you think about, this is one stretch of river where there's a very vulnerable and interesting biological resource. And what are the ethics, I guess, behind just letting this one place be without floating? And there's plenty of other rivers all around where people can do this. But we're just taking the approach where we want to do this collaborative conservation. But there's a group of people in the stakeholders that are getting more concerned. And as we see as we see the number of active nests this year, it's going to probably get more concerning toward the Slate River Working Group. And I don't know what they're going to do, but our data is just there to kind of support the decision making. So we'll see what they do with it.
Yeah. And we may need you to go back to a previous slide here for this question. It looks like there isn't much of a relationship between the number of disturbance events in a particular year and the reproductive success. Is that correct?
I think I want to go back.
So is it possible that the birds are tolerating a certain amount of disturbance, still having reproductive success? So
is this the slide that you're interested in?
Number of disturbance events, or maybe it's looking at across two slides.
Yeah, that's a great point, and I think it's fairly accurate. That the biggest thing that we're seeing is just the change in the number of active nests, but those nests that are present are doing pretty well. And you look at these number of chicks in the nest, and then the number that fledge. We had 41 out of 47, we had 43 out of 45, 42 out of 46. Those are in the 80 and 90% 100% here in 2018. And when you look at the literature, this is pretty good reproductive success. A lot of heronries are showing much lower than that, around 50%. So something is going right with this site, but there's also at the same time something not going right with the site.
Yeah.
So yeah, those that, I think my hypothesis on this that I came to is that what happens to these fledged chicks, right? Like once they leave, we don't know where they're going and we don't know what happens to them in that first year, this is the most vulnerable time in a heron's life. Once you fledge from the colony, you are now on your own pretty much to feed, and you're not an experienced fisher bird, or they eat a lot of other things, amphibians and rodents and so on, but they're not experienced hunters, and they may also be leaving the nest in poor body condition. And just because they survive and they fledge, it doesn't mean they're in good body condition. And based on the number of disturbances to the adults and their challenge of provisioning food, we could have chicks that are leaving the nest in poor body condition and then having poor overwinter survival. So maybe the mechanism driving the reduced nesting numbers from year to year is the number of juveniles that are actually surviving and then coming back to the colony site the next year. I don't have any data to support that, but that's just a hypothesis.
Yeah. Next question, could this study be generalized to other colonial water birds and scaled up to something academically organized, managed and implemented by volunteer field workers? Something like a more comprehensive version of the now defunct Colony Watch?
Yeah, that's what was coming to mind as you were reading the question, is that, the Bird Conservancy of the Rockies and the organizations before them tried to do this. They weren't really doing it in a comprehensive way. I think one of the challenges I see with that is that, at least with the historical Colony Watches, that it wasn't a very robust data set, that someone would go out once a year and count the colony. And that, at least that's the data I saw for the great blue herons at Slate River. So you would need to create something a little bit more robust where you're looking at the colony multiple times during the year to get a better sense of the sizes of these colonies, but then adding a whole nother dimension to it if you're really trying to look at how human activities are impacting these, which is a little trickier and definitely takes a lot more time. But it's certainly something that citizen science could address, and there'd need to be a pretty good coordination end to it.
I'm going to take two more questions and then we'll let you go. But are the nests that are doing well further or more proximal to the water? In other words, are there correlations to water and disturbance proximity? I know you said 300 meters. Is that, pretty much when you get inside that 300 meter range, you're going to disturb the nests?
Yeah, on this, can you guys see this photo?
Yeah.
This is the lower colony, and if you go and you can see that blue line, it goes right underneath those nests. Here's the upper colony, here's the blue line going right in front of those nests. This, in the lower colony, it's going right behind those nests. But from this lower colony to the road, that's about 300 meters. And so, most of the activities are happening over here and nothing's really affected. The only thing that's really up, you can see how complex this is, and nobody really goes out there to go fishing. Just for nobody's out there. The only people that are out there are people on the river, especially 80% of them are on sup boards.
Yeah.
But there's absolutely no spatial distance separating this river recreation from the colonies. And you can see both of the colonies are right above the river.
Yeah.
So, yeah, so we don't have any way to assess, if the colony was farther away from the river, if it would be less disturbance there, because both the colonies are right on the river.
So last question, any ideas of the return rate of the same birds versus new birds nesting? Is any banding going on to help you understand that?
Yeah, geez, we have so many questions and mysteries around this study and things we thought about. Wouldn't it be cool to put some some kind of radio telemetry on these birds, but I haven't really figured out a good way of doing that. Like it'd be easy. The easiest thing to do would be to go into these nests and put telemetry collars on the chicks. But of course, these nests are about 60 to 80 feet up in the air in some fairly precarious trees. And we're very sensitive about disturbing the colony. So we don't want to really do that. So we'd somehow have to catch herons off the nests in their foraging habitat to get any of that kind of information. So we haven't gone that far into this, but those are the questions that we're really interested in, is like, what happens to these birds after they leave, and how many come back, and are we getting the same birds? Clearly, we're getting similar birds coming back year after year because there's this site fidelity, and this colony has been around at least since the 1990s. So somehow the birds are returning to the same site, but how many and which ones, we just really don't know.
Yeah. Always more to learn.
Yeah.
And I do know that Crested Butte has a reputation of being a great stand-up paddleboard site. It is really known for that. So
yeah, Crested Butte has a really interesting dualistic personality, because at one hand, I mean, this is a great example. The Slate River wetlands are this beautifully protected area, and they have the Crested Butte Land Trust, and they've put millions and millions of dollars into protecting land and protecting natural resources. At the same time, this is one of the most recreation avid communities in Colorado, and everybody's super healthy and everybody wants to be out and do stuff. And it really is an interesting tension, because how much are people willing to not recreate to protect their land? And even this land that the Crested Butte Land Trust owns, they have a dual mission to allow recreation, but also to protect the natural resources. So how do you do that? And I think it's not really possible in this case. You've got to make that separation in space between the humans and the birds.
yeah. Well, thank you very much. We've kept you a lot longer than we thought, but we had so many questions, and it's really a fascinating story. So thank you so much for sharing.